Very good morning, dear students. So, we have been discussing unit number 6, that is fatty acids and lipids, and the previous class we have highlighted the basics of fatty acids and lipids and what are the major nomenclature of the fatty acid and a lipid. And we also discussed how fatty acids are classified. They are mainly classified into three groups, that is simple lipids or homolipids, compound lipid or heterolipids, and derived lipids. Now in this topic we are going to discuss the simple lipids. So the next topic mentioned in your syllabus, that is acyl glycerols and waxes. So these are simple lipids. And before we are going to discuss the details about acyl glycerols and waxes, you should know what is simple lipids. So a simple lipids is a fatty acid esters of different alcohol and which carries no other substance, which consists of only fatty acid esters of different alcohol. And these lipids belongs to a heterogeneous class of predominantly non-polar component which are insoluble in water and soluble in non-polar organic solvent like chloroform and benzene. And as I said, these are esters of fatty acid with various alcohol. And these simple lipids are mainly consisting of fats and waxes. Okay, the fatty acid and waxes. So most important types of fats are triglycerides. Okay, so the fats they are the esters of fatty acid with a glycerol, and oils are fatty acid in liquid state, and fats are also the lipid, they are the solid state. Okay, so mainly the fats are known as triglycerides because all the three hydroxyl groups of glycerol are esterified, hence it is known as triglycerides. Okay, all the three positions of the hydroxyl groups of glycerol is esterified with fatty acid, thus it is known as triglycerides. Then waxes are the solid esters of long chain fatty acid. Okay, so examples are palmitic acid, which are aliphatic, okay, or alicyclic or high molecular weight monohydric alcohol. They are also has alcoholic derivative. They are either monohydric or cyclic or aliphatic component. Then waxes are water soluble due to the weakly polar nature of the ester group. Their polar group is very weak in nature, and that is hence they are water insoluble. And the major types of fats are acyl glycerols. Okay, so acyl glycerols are otherwise known as triglycerides or triacyl glycerols. Why it is known as triacyl glycerols, I have already discussed. Then these triglycerides are the most abundant of the all forms of lipids. Okay, most abundant lipid present in the organism. 98% of body's dietary lipids are triglycerides. Okay, then the only remaining 2%, that is phospholipids and cholesterol, is present in our body. And these are the major storage or deposit fats, that is in plants and animal cell. Okay, and these are the major and deposited fats that is present in plants and animals, and they are commonly absent in the biomembranes, and these are non-polar, hydrophobic molecule. They contains no electrically charged or highly polar functional groups. Okay, they are non-polar and hydrophobic. Obviously, they are not having the charged molecule in their structure. In animal, the fatty acids are deposited in a particular cell that is known as fat cell or adipocyte. Okay, so that is commonly present in the organism, and the adipocytes, the fats are stored in the form of triglycerides. Okay, and also some droplets. The adipocytes are the abundantly found in that which under the skin. Commonly that is under the skin there is a thick layer, and in the abdominal cavity and the mammary glands. Okay, adipocytes are abundantly found under the skin and in the abdominal cavity and in mammary glands. Then the triglycerides or triaceroglycerides has so much importance in the organisms. Okay, so these are the triglycerides are stored in quantities to supply energy that is needed for our body. Then apart from that, they are better adapted than carbohydrate, especially glycogen, to serve storage form of the energy. Okay, the fats have more capability to store the energy than carbohydrate, and the triglycerides are stored in large amount and they provide double amount of energy than carbohydrate. I had already told you 1 gram of carbohydrate can provide approximately 4 to 4.5 calorie per gram, but in the case of lipids or fatty acid, double the amount of energy, such as 9 to 9.2 to 9.4, that much amount of energy 1 kg of 1 gram of triglyceride can produce, and they are digested more slowly than the carbohydrate. Okay, and they have greater satiety. Okay, so the greater satiety value than the carbohydrate. Satiety value in the sense they are able to maintain the state of the system. Okay, that can be in solid or they can be in the liquid as well. Then you can see here some pictures. So especially in the Arctic and Antarctic animals, okay, such as the blue whales or whales, okay, seals and walruses and penguins. Okay, so they contain lot of triglycerides, and these triglycerides, okay, these organisms are able to make use of these triglycerides into two main way. First one, they act as the storage energy form. Okay, they act as the storage energy form to serve energy storage deposit. Second one, in Arctic and Antarctic region, the climate conditions are so tough to animals to or organism to exist. Okay, so it provides an insulation against very low temperature. In Arctic and Antarctic region, the temperature is very low, even that is close to zero. Okay, so that is why this organism is having an insulation around in their skin that provides the adequate temperature. The organisms such as whales as well as penguins, etc. Okay, then the triglycerides in a normal human being, okay, healthy human being, where a person's weight is approximately 70 kilogram, 10 to 20 percentage of the body weight is lipids. Okay, that is mainly triglycerides. So I will be using TAG. TAG stands for triglycerides. Okay, and this triglyceride found in all organs of the body, particularly in the adipose tissues. Okay, and the droplets of triglycerides mainly represent more than 90% of the cytoplasm of the cell. Okay, the cytoplasm also has 90% of more than 90% of cytoplasm. They are lipid droplets. Okay, and these triglyceride tags stored in water free state in the tissue But in the case of carbohydrate, they are stored in water bound state. Okay, so the lipids are non-hydrated, but fatty acids but carbohydrates are hydrated. We have already discussed the salient features of carbohydrate, which are hydrated. Okay, and these triglycerides are esters of glycerol with 3 fatty acid molecule. Okay, so I have already told you most of the fats and lipid upon hydrolysis they produce fatty acid and glycerol. So here you can see CH2OOCr1, CHOOOCr2, CH2OORCr3. Okay, where R1, R2, R3 are the various fatty acid residues, and this stabilized by an ester linkage. Okay, so CO or linkage is known as ester linkage. This is the glycerol residue. That's what I told you. A part of the glycerols are stabilized by fatty acid, hence it is known as triglycerides. Okay, so this is the general structure of triglycerides. And this we have already discussed how triglycerides are produced. So we have already discussed this one, glycerols combined with the fatty acid, and what will happen? CH2OH, CHOH and CH2OH. Here you can see then this lipid number 1. Okay, that is R1. That means to say CH3, CH2, 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 COOH. Okay, similarly second fatty acid and third fatty acid. Okay, and here you can see in hydroxyl group the OH, and here the carboxylic acid side chain H will be combined to form a water molecule. Water molecule will be produced, then it will convert into diglycerides, then the next water molecule will be released out. Okay, that will be monoglycerides. Okay, then from there it will be converted, it will be released the next water molecule. Okay, from this side then it will be glycerol and fatty acid. Okay, so that is what I told you, it will be that is what is happening. Okay, glycerol and fatty acid. So during the formation what will happen? The just opposite process will happen. Okay, so here you can see this will be combined to form this structure. Okay, this structure, that is COO bond will be produced here, that is esters. Here also esters, here also esters. Okay, so that is ester linkage. So this is what I told, when triglyceride undergo hydrolysis, this produces, first it will produce diglycerides, then it will produce monoglycerides, finally the complete fatty acid and glycerols. Okay, so this is the structure. Okay, so this can be represented like this also, the glycerol, the full structure, and you can write it the zigzag line, that is a alkane side chain. Okay, this fatty acid residue, alcohol residues here. Okay, here COOH group is also here, so interaction is happening due to this particular end. Okay, then there is a strong ester linkage you can see. Okay, and this will be remains like this. This is triglyceride. So when triglycerides undergo hydrolysis under the in presence of lipases, 3 molecules of fatty acid and glycerols will be produced, plus the water molecule will be involved. Okay, so that is the formation of triglycerides. Then these triglycerides are also optically active. They are able to form isomerism. Okay, so isomerisms are the compounds, they are having same structural formula but different spatial arrangements. Okay, so when these molecules, when you look here, you can see carbon number 1 and carbon number 3. Between 1 and 3 there is carbon number 2. This acts as the chiral carbon atom, because this is one residue, the second residue, this third residue and fourth residue. 4 different positions of carbon atoms are fulfilled with the valencies are fulfilled with 4 different groups or atom. Thus it is a chiral carbon atom or asymmetric carbon atom. Okay, and what I am trying to tell is these triglycerides can exist both the dextrorotatory form and levorotatory form, that is both D variety and L variety like a carbohydrate, and the naturally occurring form is L variety. Okay, here you can see the D isomers where all the carbohydrate, that is fatty acid chain along with the ester linkage are the same side. Okay, So it is first carbon atom OOCr1, second one OOCr2, that is OOCr. This is cis configuration. Okay, but in the case of this is known as a D conformation. Okay, but in the case of L conformation, you can see the chiral carbon atom. This is COO R1 and this is COO R2, just opposite. Okay, and this is there is a difference between here and here. Okay, so that is just a mirror images actually. Okay, so that is what I told you, that is they're able to produce a isomerism. Okay, and this L variety are the common type of glycerides. Then how triglycerides are classified? The triglycerides are classified based on whether the fatty acid chains are same or different, whether based on the similarity or dissimilarity of the fatty acid residue that is commonly present. Okay, they are classified into two groups. One is simple or symmetrical triglycerides, or mixed or asymmetrical triglycerides. As the name indicates, simple or symmetrical triglycerides are which consisting of only one type of fatty acid in all the three positions of the hydroxyl group of glycerol. So all the alpha, beta and alpha dash or beta dash, whatever it is, that is produced same type of, that will be having same type of fatty acid. Example, tri-palmitin, tri-stearin, tri-olein. Okay, here we can see structure of tri-stearin, that is known as tri-stearoyl glycerol. Okay, so what is this? This is the CH2COOR1. Okay, instead of R1, all the R1, R2, R3, all the three position you are having what? You are having stearic acid. Okay, hence it is known as tri-stearin. Similarly, tri-olein, R1, R2, R3, C17H33COOH. So all the three structure you can see this is the same, C17H33, C17H33, 317H33COOH. Okay, that is oleic acid. Hence they are known as a tri-olein. These are simple triglycerides. So simple triglycerides are the one of if the all the fatty acids are the same kind. Okay, so only one type of fatty acid, that is known as a simple triglycerides. Then mixed or asymmetrical triglycerides, which contain two or more different types of two or three different types of fatty acid in their side chain. Say for example, oleodipalmitin and oleopalmitostearin. Okay, here you can see oleodipalmitin. Here it is one place. Here you can see this is D form, but this is L form. Okay, so CH2 C17H33. Okay, you have already known that is oleic acid. Okay, here one oleic acid residue, but in second fatty acid you see R2 COO, that is C15H31. Here it is C17H33. Here it is C15H31. That is palmitic acid. Here also C15H31. Okay, that is palmitic acid. Two palmitic acid residues and one oleic acid residues. That is known as a oleodipalmitin. Okay, oleodipalmitin. Okay, then or oleopalmitostearin. In this case, the R1, R2, R3 are fulfilled with here it is a C17H33COOH, that is oleic acid. Then C15H31COOH, that is palmitic acid. Then C17H35COOH, that is stearic acid. Okay, so that is what oleopalmitostearin. Oleic acid is there, palmitic acid is there, and stearic acid is there. So that is what I told you. This is a mixed or asymmetrical triglycerides. Then these are some of the common types of the fatty acid, their composition, and that is the composition and percentage of total fat which is present in the plant and animal cells. Okay, in the case of animal fats, example butter fat, they are highly saturated. The percentage is 60% of the butter fat is saturated, then 36% is monosaturated. Only one type of monosaturated, you know that is hydrogen bond. Only one type of double bond is present. Then polyunsaturated. Okay, they have mainly 4% of polyunsaturated. They can exist most of the case they can exist in the saturated form, then comes monosaturated form, then very rarely they can exist as a polyunsaturated form. Okay, only 4%. Then pork fat, similarly 59% they can exist as saturated fatty acid, 39% they can exist as a unsaturated fatty acid, and only 2% they can exist as polyunsaturated fatty acid. Okay, so beef fat, that is 53% they can exist as saturated fatty acid, 44% they can exist in the form of a monosaturated fatty acid, and 2% they can exist as a polyunsaturated fatty acid. Similarly, chicken fat, 39, chicken is generally, that is very less content of a saturated fatty acid, especially that has only 39% of saturated fatty acid, but here the monosaturated fatty acid is more, it is 44%. Okay, but polyunsaturated fatty acid is 21%. Here you can see almost the the percentage of different types of fatty acid is almost more or almost balanced in the case of chicken fat. So what I am trying to tell is, in the case of animal fat, more percentage will be saturated fatty acids. That is what I told you. Animal fat, they are saturated in nature. They are they have a less chance of finding a double bond. But there are few examples are also there. But in the case of plant fat, there is just opposite. Then olive oil, okay, they are having only 20% they can exist in the form of saturated fatty acid, 26% they can exist in the form of unsaturated fatty acid, 54% they can exist as a polyunsaturated fatty acid. Similarly, corn oil, 15% is saturated fatty acid, they can exist as 31% they can exist as monounsaturated fatty acid, and 53% they can exist as a polyunsaturated fatty acid. Then similarly, soybean oil, okay, 14% saturated fatty acid they can exist, then 24% they can exist as monounsaturated fatty acid, and 53% they can exist as polyunsaturated fatty acid. That is why plant oils, you can see they are rich in polyunsaturated fatty acid. The number of double bonds are very, very high, okay, and they are unsaturated in nature. But in the case of animal fat, they are saturated in nature. Then what are the main difference between the animal and plant fat? What is the difference between animal fat and plant fat? In this table you can see in the case of animal fat, they are relatively rich in saturated fatty acid, as I said. Okay, so generally they have C16 and C18 acids. C16 carbon number 16 and carbon number 18 residues will be there. But in the case of plant, that is relatively rich in unsaturated fatty acid, especially polyunsaturated fatty acid. Here you can see the percentage that is very, very high in the case of a plant fat. Okay, then the animal fat solid at a ordinary temperature, but in the case of plant fat, they are liquid at a room temperature. Okay, then animal fat have a low iodine number. We will discuss what is iodine number. And the plant fat have a high number of a high iodine number. Then these animal fats have a usually a high Richard Meisel number, and the plant fatty acid have a low Richard Meisel number. So what is this Richard Meisel number? That is an index of the volatility of fatty acid. Okay, the volatile component of a fatty acid can be represented in the form of Richard Meisel number. But in the case of animal, that is high. In the case of plant fat, that is very less. Then these animal fats are stored mainly in liver and bone marrow, but in the case of plant fat, they are mainly stored in seeds and fruits. Okay, then the animal fat, their oxidative rancidity observed more, but plant fat, the oxidative rancidity is observed less. So what is this oxidative rancidity? When fatty acid is reacted with the oxygen, the triglycerides will be undergone hydrolysis, and what will happen? Either with the help of water, or that is known as hydrolytic rancidity, or with the help of oxygen, they'll completely broken down, and it will undergo certain bad smell, that is a rancid smell. Okay, the fatty acid rich food item undergo hydrolysis or oxidation, they become what will happen? Especially in the oxidation, they will they convert the smaller fragment and they will acquire a characteristic very foul smelling, that is known as oxidative rancidity. Okay, in the case of animal fat, that is more, but in the case of oil, that is less. Okay, example of this animal fats are such a kind of oxidative rancidity will be commonly happening: the beef fat, butter fat, tallow and corn oil. Okay, but in the case of plant fat, olive oil, castor oil, soybean oil, etc. Okay, so these are the main difference between animal fat and plant fat. So these are about a triglyceride. The second part of the simple glycerides are the simple lipids are waxes. So the waxes are the material of a, generally waxes, they are the material of the honeycomb, that is beeswax. The honeycomb is made up of beeswax. Okay, we will see that. Okay, then what are waxes? These are the esters of long chain saturated and unsaturated fatty acid with long chain monohydroxy alcohols. Okay, the waxes are the esters of long chain saturated and unsaturated fatty acid with the long chain monohydroxy alcohols. Okay, then the fatty acids are commonly ranged in waxes between C14 and C36, very long chain. Okay, and the alcohol which are ranges from C16 to 3 C36. The fatty acid portion, they ranged from C14 to C36, and the alcohol portion, that is ranged from C16 to 3 C36. Okay, in vertebrates, the waxes secreted by cutaneous gland, which are the protective coating, that is commonly present in the protective coating of the skin. Okay, and this is commonly present in hair, wool and fur, which are coated with the fats. Okay, and the birds, you can see beautiful birds. Okay, they secrete lot of waxes to make their feather. Okay, water repellent. Okay, so here you can see when this kind of water fowls, okay, when they swim in water or they fly, okay, immediately their feather, okay, the body feather is completely repellent of water. Okay, due to the presence of this waxes, a very thin waxy coatings are commonly present in the feather of these particular birds. Okay, so that is the important aspects. And there are a lot of examples for waxes. So most of the waxes are a mixture of the mixture of esters. Okay, the sperm whale wax, that is known as spermaceti, and beeswax, which are composed mainly of palmitic acid, which are esterified with either hexacosanol or triaconitanol. Okay, so hexacosanol and triacotanol. Okay, so that is commonly present. So sperm whale wax this is known as a sperm whale. Okay, in this region of that particular wax, that is known as there is a particular region called a spermaceti organ. This I can show you here in a bigger way. Okay, the same thing. Okay, this region, okay, of this particular whale, they are having spermaceti organ, and this spermaceti organ consists of lot of waxes, that is known as spermaceti. Okay, that is commonly present in the case of sperm whale wax. Okay, and this consisting of what? These two, this palmitic acid, that is esterified with the either with this main component or with this component. Okay, and the sperm whale wax. Okay, and similarly I told you the beeswax also. Here you can see the honeybee, they will be network, they will be made up of their wax. Okay, this wax is very beautiful, that is completely wax actually, that will be composed of palmitic acid, that is esterified with hexacosanol and triacotanol. Okay, so and this sperm whale wax is also rich in acetyl palmitite. Okay, this is very rich in acetyl palmitite, and bee wax is primarily consisting of myricyl palmitite. Okay, myricyl palmitite. And I told you how this is produced. When palmitic acid is interacted with the hexacosanol, that will be produced this sperm whale vaccine. Okay, CH3 CH2 14 times COO and CH2 CH2 24 times CH3, that is hexacosanil palmitite. So that is known as spermaceti. Okay, this is an excellent example of a wax that is commonly present in the universe itself. Then again you are having another wax is known as a Kannur wax. Okay, Kannur wax, that is the hardest non-wax, that is consisting of mainly fatty acid esterified with tetracosanol or tetraacon-tanol. Okay, so tetracosanol or a tetratriacon-tanol esterified with the fatty acid. Okay, and this fat that is the waxes are from, that is that from the obtained from the conifers contain polymers formed by the ester linking mainly the omega hydroxy acid, especially juniperic acid. Okay, especially juniperic acid. Okay, and these waxes are commonly, certain waxes are commonly present, they have a characteristic odor. Okay, so due to the presence of hydroxy acid, okay, in the form of lactones. So certain waxes they have a characteristic smell, that is due to the presence of hydroxy acid in the form of lactone. An excellent example is umbritolide. Okay, umbritolide, a wax that is extracted from the seeds of lady's finger. Okay, the lady's finger, you can see, if you cut, you can see these seeds here. Okay, the seeds of lady's finger, you can see this umbritolide. There is a characteristic wax, okay, that has a musky smell. Okay, and this, because of this particular component, this seeds of lady's finger has got a musky smell. Okay, and similarly the lipids of marine organisms such as starfish. Okay, here you can see starfish or squid. Okay, squid or shark, they have a fatty alcohol, and that helps to maintain their body in a proper way, and that has long chain alkyl ethers of glycerol. Okay, and that is very much essential for their survival system. So these are some of the typical example of waxes. Okay, then comes the property of waxes and their main uses. The waxes are usually they are inert. Okay, they are inert due to their saturated nature. The waxes are saturated, that is they are rich in saturated fatty acid. Okay, and the hydrocarbon has a saturated nature. Because of that reason, usually they are inert in nature. Then they can split slowly with the hot alcoholic KOH. If you apply the alcoholic KOH, they will be able to convert into smaller form, or they can split. Okay, they are insoluble in water, and they are highly resistant to atmospheric oxidation. Okay, these waxes are resistant to atmospheric oxidation. They are highly insoluble in water, and these are usually polishing. These are used to polishing furnitures and automobile, and also wax coated paper that is commonly used to wrap the perishable food items like biscuits, cake, etc. Okay, so such a kind of a waxy, when you are opening certain biscuits, cake, you can see there is a very waxy, a very thin coating paper that is made up of this kind of waxes. Okay, simple lipids. Then another important example is there are lot of other waxes like like spermaceti. Okay, lanolin. Okay, that is lamb's wool. Okay, that is obtained from lamb's wool. Okay, beeswax, that is obtained from honey comb. Okay, I have already told you about this. Kannur wax, that is obtained from a Brazilian palm tree. Okay, this is commonly obtained from a palm tree. And spermaceti oil obtained from sperm whales, okay, which are widely used for, okay, this lanolin, beeswax, and carnauba wax and spermaceti oils are commonly used to manufacture lotions, ointments, and polishes, etc. Okay, so waxes has lot of application in the industry as well. So this is all about acyl glycerols and waxes, triacyl glycerols and waxes. So with this we can conclude. So today we have discussed about the introductory aspects of triglycerides, mainly simple lipids. We have discussed what are simple lipids. Then we have discussed about acyl glycerols, mainly triacyl glycerol, why it is known as triacyl glycerols or triglycerides. Then we have discussed about the overview of triglycerides, their structure, how triglycerides are produced, formed, what are the isomerism of triglycerides, then how triglycerides are classified depending upon the nature of the fatty acid chain attached, then the what is the fatty acid composition of important animal and plant fat, and what are the major difference between animal and plant fats. Then what are waxes, what are the major examples of waxes, and what are the salient features of waxes, and what are the uses of waxes. Okay, this is all about this acyl glycerols and waxes. So these are the main textbook along with the principles of biochemistry by Leninger. You can refer biochemistry by biochemistry by J.L. Jain. That is a very good textbook. Okay, so thank you dear students for watching this particular class. Okay, if you are having any clarification you can ask me. Have a great day.